Welcome back, dear students and friends. We have started a discussion on chapter number 8, the cell, the basic unit of life. We have discussed the prokaryotic cell and we have discussed the many organelles of eukaryotic cell. Students, today we have to learn the another organelle of eukaryotic cell that is found in a eukaryotic cell. And today we have to learn the cytoskeleton. Okay. What is a cytoskeleton? Okay, you know the skeleton. Skeleton, that means in our body the skeleton that consists of bones. It consists of cartilage. Okay, this is the skeleton. Okay, that is responsible for a support and the movement in our body. Similarly, the skeleton of cyto means the skeleton of a cell. Cyto means a cell. Okay, so the skeleton of a cell, this is called as cytoskeleton. Okay, so what is a cytoskeleton? The cytoskeleton, it is the filament-like structure. They are the filament-like structures present in cytoplasm of a cell. Is called cytoskeleton. It's called as cytoskeleton. Clear? What is a cytoskeleton? Cytoskeleton means the filament-like structures that is present in the cytoplasm of a cell. This is called as a cytoskeleton. Okay? What is the functions? What are the functions of this cytoskeleton? The functions of cytoskeleton. Okay? The first function of this cytoskeleton is it gives shape of a cell. You know that there are different varieties of shapes of a cell like spherical, oval, discoid or many others. Okay, this shape, the shape of a cell is going to be maintained due to a cytoskeleton. Okay. Second, it gives a support to a cell. Okay, it provides a support to a cell. Okay, so this is the second function. And the third function of this cytoskeleton is that it is responsible for motility. That means a movement. These are the main three important functions of a cytoskeleton. It gives a shape of a cell, it provides a support to a cell, and it is responsible for a motility. Okay. Here there are three types of the cytoskeleton that is found into a cell. What are the types? Types of cytoskeleton. Okay, there are three types. First one is called as microfilament. Second one is called as microtubule. And the third one is called as intermediate filament. These are the three different types of a cytoskeleton you can find into a cell. I again repeat, which are they? Microfilament, microtubule and intermediate filament. Most of them, they are made up of protein. Like for example, remember the microtubule is made up of a tubulin protein. Which protein? Tubulin protein. Okay, so this is, it's called as a microtubule. Intermediate filament, it is the strongest and the most durable protein. It is the intermediate filament, it is the strongest and most durable protein. Okay, so these are the different functions, but we don't have to deal it in detail according to a syllabus. Even also these three names are not given in syllabus, but they are important from a neat point of view, so you have to prepare it. What are the three different main important types of a cytoskeleton? Microfilament, microtubule and intermediate filament. Microtubule is made up of which protein? It's made up of tubulin. And which is the strongest and most durable filament? It is the intermediate filament. Okay, so this is all about the cytoskeleton. Okay, skeleton of a cell, a filament like structure that's found in a cytoplasm of a cell, and these are the functions. Give a shape, provide the support, and it's responsible for motility. Okay, so this is about a one organelle you can find into a eukaryotic cell, that is cytoskeleton, a sweet and short. Now we will move ahead with another organelle, and that is cilia and flagella. Okay, another organelle you can find into a eukaryotic cell is a cilia and flagella. So now let's discuss cilia and flagella. What is a cilia and flagella? Cilia and flagella, they are hair-like projections from the surface of a cell. Okay, what they are? They are hair-like projections which occurs from the surface of a cell. They are called as cilia and flagella. Okay. Now, this cilia and flagella, what's the difference between this cilia and flagella? Both are hair-like projections that occurs from the surface of a cell. But cilia, they are many numbers. But the flagella, they are one or two. Okay, the flagella are one or two. Okay, next, the another difference between cilia and the flagella is that the cilia, they are short, whereas the flagella are long. Okay, they have a common functions. They are responsible for motility and the movement. So this is the difference between the cilia and the flagella. Both are hair-like projections that arise or generated from the surface of a cell, but the cilia are many in numbers and they are shorter. Okay, whereas the flagella is one or two and they are long. The cilia is responsible for movement of cell as well as it is also responsible for movement of cytoplasm. Flagella is responsible for movement of a cell. Okay, so this is a difference which exists between the cilia and the flagella. Okay, now cilia and flagella both have a mostly common structure. You can say a common structures. So we will discuss the cilia and flagella as a common structure. Whatever the difference has been there between the cilia and flagella have been discussed over here. Otherwise, structurally the cilia and flagella are similar. Okay, so what it contains? If you see the cilia and flagella, they are the tubular structures or the filamentous structures. Okay, these filamentous structures have a central axis. So if you take a section of this, you can find a circular shape. Okay, the center of that axis, that is called as an axone. So I am drawing just an axone. Okay, so remember, this is what is called as an axone. What is axone? Okay, the axone means central axis. Okay, central axis of cilia and flagella. This is called as axone. What is called as? It's called as an axone. Now, if you see the structure of this axone, central axis, then what you are going to find? You are going to find there are pair of or doublet microfilament. They are present on the periphery. This is a periphery and this is a center. In the periphery, you can find 9 pair of doublets. This is a doublet. I am drawing a doublet. This is a doublet. 2. Okay. So, how many? This is a one doublet. How many doublets are present? There are 9 doublets are present. So this is a 1. This is a second, third, fourth, fifth, sixth, seventh, 8, 9. Okay, 9 doublets are present. I again drawing because it's an unequal distribution, so I am again drawing, so you can accommodate all the 9s. Okay, I again draw. This is one doublet, this is another second doublet, this is third doublet, it's a fourth doublet, this is fifth, this one is sixth, this one is 7, this one is 8, this is 9. So 9 doublets are present on the periphery. Okay, they are present on the periphery. Okay, they are present in a pair. They are present in a pair. Pair means 2, all. So how many pairs? 9 pairs are present. And in the center, there are 2 individuals are present. 2 individuals. They are not in a pair. They are 2 individuals. Such kind of arrangement, it's called as 9 plus 2 arrangement. What it's called as 9 plus 2 arrangement. What's a 9 plus 2 arrangement? 9 plus 2 means in the structure of cilia and the flagella, there is a central axis called as an axoneme, which contains 9 doublets in the periphery, okay, or 9 pairs of filament in the periphery and the 2 individuals in the center. Such kind of arrangement is called as a 9 plus 2 arrangement. Okay, these two which is present in the center, these are the 2 which are present in the center. They are covered or surrounded by a sheath. They are covered or surrounded by a sheath. Okay, and these 9 doublets, they are attached to a sheath via radial spoke. So this is a radial spoke by which they are attached. Okay, they are attached to a central sheath. Okay, so by this radial spoke, you might have seen the tire, a radial tire. Okay, from this radial tire, the spokes are going to be generating from the central axis. Same type of arrangement you can find it in. Or you might have seen the arrangement of a cart wheel. In the cart wheel, in the wheel there is a center, and from this center the spokes are going to be arranged. Okay, so these spokes which are attached to it. Okay, so such kind of arrangement can be found in the structure of cilia and the flagella. Okay, so this is about a structure of cilia and flagella. Again repeat, what kind of structure? The central axis is called as an axoneme. The axoneme, it consists of 9 doublets which are arranged in the periphery, and in the center there is a presence of 2 individual filaments. Such kind of arrangement is called as a 9 plus 2 arrangement. The 2 filament which is present in the center, they are covered by a sheath. This is a sheath covering. Okay, you can say this is a sheath or covering in simple language. The doublets which are present in the periphery that attaches with a sheath, okay, radially and form a spoke-like arrangement. So this kind of arrangement can be found in the structure of cilia and the flagella. Okay, remember the different types of paramecium, it possesses the cilia. Paramecium, remember, I will tell you the example. Okay, examples have not been given in the book, but the paramecium, it contains the cilia. Euglena, E-U-G-L-E-N-A, it possesses the flagella. Okay, and the bacteria possess both cilia and flagella. But remember, the structure of flagella that is present in a prokaryotic cell and the structure of flagella in the eukaryotic cell, they are different. Both are called as a flagella, but the structure of cilia and structure of flagella in prokaryotic cell and the eukaryotic cell, they are different. Here we have discussed the structure of cilia and flagella in eukaryotic cell. Okay, so this is about the second organelle what we have discussed. It is cilia and flagella. The first one is the cytoskeleton, the second one is cilia and the flagella. Now we are discussing the last organelle of this today's session, and that is centrosome and centriole. Here we are discussing the centrosome and centriole. Okay. See, centrosome and the centrioles, they are arise from the basal body. Okay. So the centrosome consists of tubular structures like this. This is a one tubular structure, tube-like structure. Okay. And this is the another tubular structures. So the centrosome consists of two tubular structures. Okay. And these two tubular structures, they are arranged perpendicular to each other. They are arranged perpendicular to each other. Okay. So the individual, this is called as a centrosome. It consists of two filament structures. Okay. And how they are going to arrange? They are arranged perpendicular to each other. So the angle between them, this is the angle between them, is 90 degree. Because they are arranged perpendicular to each other. So this is an important question which is going to be asked. What is the angle between two centrioles? Because this individual one is called as a centriole. Okay. So in short, you can say that the centrosome is made up of centrioles and the angle between them is called as a 90 degree. Okay. So this is called as a centriole. Okay. Now the arrangement of this centriole is also similar to a cilia and flagella. Okay. This is the arrangement is also a cilia and flagella. Okay. How they are? They are the 9 triplets. In the cilia and flagella, 9 doublets. Okay, means 2. They are arranged in a 9 numbers in a periphery. Here 9 triplets. So this is a 1 triplet. This is second triplet. Similarly, this is a third one. This is the fourth one. Fifth. Sixth. 7, 8, and this is a 9. 9 triplets are present, and in the center, this is the presence of a center. This center is called as hub. Okay, so the 9 triplets, they are present in a periphery, and in the center there is a presence of a hub. Okay, these 9 triplets are connected with a hub like this. So it also forms a same kind of a arrangement like a spoke. Radial spoke like a arrangement is going to be formed. Okay, and here the adjacent triplets are also connected. Adjacent triplets are also connected. Clear? These are the adjacent triplets. They are also connected in the structure of a centrosome and the centriole, or you can say specifically a centriole. Such kind of arrangement you can find. I again repeat, what is the arrangement into a centriole? In a structure of centriole, 9 triplets, 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 triplets, they are present in the center, and 9 triplets are present on the periphery, and on the center there is a presence of hub. These triplets, they are attached to a central hub and form a radial spoke-like arrangement. Even these triplets, they are also connected with the adjacent triplets. Okay. Remember, there is one more important question regarding to a NEET point of view. The angle between two adjacent triplet is 40 degree. Remember, they are confusing you. The angle between two centriole. This is the angle between two centriole. That is 90 degree. But in a one centriole, this is the arrangement of one centriole. The angle between two adjacent triplet is 40 degree. Okay, so you have to remember what are the angles. Okay, these angles are not given in book, but it is very important from a NEET point of view. Okay, so and these two centriole combine together and form centrosome, because the centrosome have a perpendicular arrangement of a two centriole. Okay, so the centrosome is made up of centriole, and this is the inner arrangement of a centriole. Okay. This centriole is only found in animal cell. Plant cell do not have a centriole. That already we have discussed it. And this centriole has an important role in a cell division process. So what is the function of this centriole? The function of this centriole that it involves a cell division process. Cell division means here it divides a cell, and by division new cells are going to be produced. So this is a function of centriole. So that's all about the structure and function of centriole. So students, today we have learned the three organelles which can be found in eukaryotic cell. First is cytoskeleton, second is cilia and flagella, and the third one is, it's called as a centrosome and centriole. All these are short, but they are important somewhere. Okay, so this is all about the today's session. We will discuss the another important topic in the next session. Till then students, please keep learning, keep watching, like and subscribe this channel. Do not forget to like and subscribe if you like the video. Okay, thank you for watching.